Oui, c'est normal qu'il n'y ait pas de son depuis la salle parce qu'on n'avait pas mis en marche les microphones encore. Mais maintenant, ça devrait marcher. Maintenant, tout le monde peut nous entendre? Parfait.
Yes, all good, all good.
Great, so we'll start in a couple of minutes.
Ouais, peut-être on attend 2 minutes.
Quoi?
Because of the misogyny in the UK. Okay. Okay, so hi everyone. So thanks a lot for those who managed to come despite the heat wave. So welcome to this morning seminar series in Ecologie-Evolution. So as usual, for those who attend on Zoom, you can report any issue, technical issue in the chat and ask your question via the Q&A tab, and we'll be able to let you ask your question at the end of the seminar. And with that, I leave it to Sylvain to introduce today's speaker.
Thank you. So hello, everybody. So we have the pleasure to host Britt Koskella for a few days in Montpellier. So Britt is an evolutionary biologist working in coevolution. So she's been doing her PhD with Kurt Lively, and then she did a postdoc with John Thompson and with Angus Buckling. So, and she's been working a lot on coevolution between phage and bacteria, and now she's pursuing this work and also working. She's very keen on working in the wild, and I think that's one really specificity that she's doing. And she's currently in the Wissenschaftskolleg in Berlin for the year for a sabbatical with Mike Boots. And that's it. So I'm going to let you talk now, Britt.
Okay. Thank you very much. So let's see. Okay, that's probably should work. Thank you very much. It's nice to be here. Nice to see many familiar faces. Hello. And to those of you on Zoom, I hope you're cool where you are. It's actually very cool here, so you're missing out if you're not. Okay. So I am just going to jump right in. As Sylvain said during the introduction, I'm a trained evolutionary biologist, and I still see myself very much as an evolutionary biologist who thinks about co-evolution. But something that has changed a lot, especially since I started my lab at Berkeley, is that we've started asking about how microbial interactions within hosts changes host fitness, changes the coevolutionary history of, well, life on Earth. And that has led to a really different tagline for the lab than I would have expected 7 years ago when I started my lab. And so now I think what brings all of us that in the group together is thinking about how core principles, how core theory in ecology and evolution, in particular in microbial ecology and evolution, can be used to increase plant health and agricultural sustainability. So fundamentally, I'm still interested in the really neat core theoretical questions, but it's fun to have this playground where by answering them, we're also able to change the way that we might do agriculture. And so when you think about the plant microbiome, you might think first about the below ground rhizosphere. And that would make sense because the vast majority of information that we have and knowledge we have about plant microbiomes has focused below ground. This is just the sort of somewhat recent Google Scholar results. If you search plant health and microbiome, the vast majority of them focus on the below ground. And I like to think about the rhizosphere. Actually, we don't think about the rhizosphere, so I don't like to think about it. But when I do, we think about it as equivalent to the human gut microbiome. So it's really, the main roles are in terms of metabolism and nutrient acquisition. And it's a sort of powerhouse where the plant turns nutrients into growth. But where we actually focus is above ground in the so-called Phyllosphere. And this is relatively understudied as a plant microbiome, but I would argue equally important, both in terms of a microbial habitat. So this is something like the third largest biome on Earth, the Phyllosphere. So if you're a bacterium that can grow on leaves or in flowers, then you have a wealth of environments to grow in, habitats to grow in on the planet. So from that perspective, it's a good microbial habitat, an important microbial habitat. But in terms of plant fitness and function, these microbial communities on leaves are equally important. And I'll spend the rest of today trying to convince you that that's true. But to be fair, although we do think about them in this way, and this is the motivation for the work, the Phyllosphere is just an awesome model system for an ecologist and for studying ecology and studying evolutionary biology. And this is why. So if you asked me what a population below ground looked like, I would have no idea where to start or finish. You know, the soil is just this continuous. In fact, people who work on marine systems have no idea how they keep their head on, because where does a microbial population begin and end in water? I don't know. But I'm very lucky because in this system, I have a hack, right? I can say this is a microbe on this leaf, and this is a microbe on another leaf, or on this plant and on a different plant. And that turns out to be very useful as a starting place for asking the types of questions that we ask. It's also easy to measure and manipulate. I'll show you how we do that. But as you can imagine, there's no digging involved. We can just pick a leaf and quantify what's growing on it. And similarly, we can spray bacteria or fungi or viruses that we're interested onto that leaf and ask how that impacts the ecological processes. And then it has an important function. And I will allude to this a little bit towards the end of the talk, that there is some role in nutrient acquisition, possibly nitrogen fixation and others. But for the most part, what we focus on is the role that this Phyllosphere community play in preventing pathogen colonization. And so I already mentioned to you that we think about the rhizosphere when we do as the gut microbiome of humans. Well, I think about the Phyllosphere as the skin-associated microbiome, because of course our skin-associated microbiome has a really important role as a barrier between us and the environment in both communicating with our own immune systems when a non-host pathogen arrives, something that should flare up the immune system, but also they're constantly outcompeting newly dispersed bacteria that don't belong on the skin. And so we think that this is very similar to how the Phyllosphere functions in plant health. And just to quickly introduce the pathogen that we mainly talk about and that I've been studying since my postdoc, where I was working on it in horse chestnut trees in Oxford, is Pseudomonas syringae. And it's a broad host range plant pathogen, which makes it very fun because there are some specific host adaptations, but really it affects over 50 species on Earth, very far upwards of that actually now, and many of them agricultural, so it has this sort of built-in justification. So the types of questions that we have been asking over the last few years is to understand how these Phyllosphere plant interactions shape ecology and evolution. We really have to start at basic principles. And so I'm thinking, how does the microbiome even get there? Right? How do you assemble a microbiome if it's so critical to function? And this is true of all microbiomes on Earth. How do you correctly assemble this from all of the microbes floating around in the soil and air? And then once they're there, what shapes microbiome composition? So what are the factors that change through time temporally? Who competes with whom? Then once we know that, we can start asking neat questions about the role that Phyllosphere plays in health and disease. And then I'll very quickly touch at the very end of the talk on how we can start leveraging these ideas in agriculture. Oops. And of course, you can't think about any of these questions without thinking about all the ways that we've messed this up, fundamentally messed this up. So much of agricultural practice has focused on reducing pathogens and not really thinking about all of the commensals. And so once you've thought about all of those assembly processes and how important the Phyllosphere and Rhizosphere microbiomes are, then you can think about how different this nice tomato plant growing in Peru might be in terms of community assembly of the microbiome from what we do in agriculture. And in particular, what we focus on are, what are we doing when we treat seeds? Are we disrupting seed vertical transmission of microbiomes? What are we doing when we put plants in greenhouses? We've now stopped all microbial transmission and dispersal from wind and rain and the like. And then what are we doing when we age structure and monoculture? Now you've put plants, I mean, look at this, right? This is a plant that's growing in a community of other plants of different ages. So that will probably means that there's an entirely different dispersal pool than something that's growing in a monoculture with an age-structured population. So in other words, we've probably messed up all of those transmission dynamics, including via seed, via wind, insects, and rain, and transmission from neighbors. But let's start and just ask whether these are even important to begin with. And so I'll give you quickly three quick vignettes about how we've been studying transmission of the microbiome, and then I'll move on to how the microbiome is shaped. So this is work led by a previous PhD student, Norma Morella, who's now a postdoc at the Fred Hutch Cancer Institute, and Shirley Zhang, who was an undergraduate researcher who's now pursuing her PhD at Cornell. And what they did was went out into the field in an organic farm and sampled tomato plants from 4 different cultivars of tomato. They then surface sterilized the fruit, fermented the fruit, collected the seeds, and simply asked, what is growing naturally epiphytically on tomato seeds? And we did 16S amplicon sequencing, and the punchline was that they're not sterile, which is not surprising. It's primarily Pantoia species. If you squint, those are mainly different species of Pantoia, and certainly less diverse than what you'd find on an adult plant. But nonetheless, there was a microbial community there on these seeds. These are across the 3 different cultivars. Well, 3 out of 4. One of them didn't work very well. I'm going to come back to this story when we get to plant function, because this was really only the beginning of the story. But for the purpose of this introduction, suffice it to say that transmission of the microbiome is happening, or is likely to be happening, through vertical transmission through seeds. And now the question is, how important is that relative to all the other transmission that happens later? Oops, so yes, we have transmission via seed. So then the second vignette I'll introduce and then come back to when we get to the plant function part of the talk. This was work led by Elijah Melferber, who's a PhD student in the lab, and he has spent most of his PhD building what we call a synthetic community. So a community of microbiota that we sampled originally from the tomato Phyllosphere, so they are naturally found there, and they're broadly phylogenetically representative of the diversity we find in the tomato Phyllosphere. But for our purposes, they're culturable, and we've now whole genome and annotated, whole genome sequenced and annotated the genomes of all of these players. And now we can control who gets sprayed onto the plant in neat combinations and ask ecological and evolutionary questions. But one of the first things we did was ran a series of trials in the greenhouse where we either sprayed juvenile tomato plants with buffer, or we sprayed them with very low densities of this synthetic community and essentially asked, is there dispersal limitation in the greenhouse? Is a tomato that's growing in regular potting soil, not sterile soil, is it getting a Phyllosphere microbiome that's sufficient for plant growth or not? And again, I will come back to a big punchline of this story later, but for the purpose of this introduction, what we found was that when we spray plants with this synthetic community, it makes a huge difference in terms of who ends up colonizing that plant. So first, if you just look at relative abundance, as most microbiome studies do, so in other words, you sequence the 16S amplicons, 16S rDNA of the bacterial community, and then you say, who's there? Not how many are there, but who are there, right? If you do that, what you find is that control plants that were not sprayed with the SynCom have a microbiome, but you see they're all lightly shaded. These are taxa that we did not spray onto the plants. And this is in a randomized greenhouse, an open greenhouse. So these are plants right next to one another. But clearly, if you don't spray the SynCom on, it's not the dominant taxa. So if you now look at those that we sprayed with, we call our synthetic community Phylostart. If you now look at the plants sprayed with Phylostart, they are clearly dominated by taxa that we sprayed on. Where it gets really interesting, though, is when you move from relative abundance to absolute abundance. And we use both qPCR as well as droplet digital PCR to estimate exactly how many bacterial cells are on the leaves. And when you do that, you notice that all of these taxa here that we didn't spray on, they're essentially not there. I mean, they are very rare on the leaf. But when you spray them on, then those taxa that you spray on actually grow to appreciable numbers. So for the sake of this part of the talk, what I want you to remember is that if you don't spray the taxa onto the leaves, they are not there in a greenhouse setting. And why that's important is it means that the greenhouse plants are going to be, excuse me, pretty depauperate of microbiota, but also that the soil, even when it's non-sterilized, is not sufficient to colonize above ground. There we go. And so another way of saying that is that in a greenhouse where plants are protected from wind and rain and bugs, they are not receiving a lot of dispersal of microbiota aerially. And just to convince you that this is a really meaningful transmission mode, I'm going to spend a bit more time on this final vignette, which is if rain and wind and bugs are important in terms of moving microbiota, then it should really matter who your neighbors are. It should matter which community you're in as a focal plant in terms of shaping the pool of dispersing organisms that arrive on you. And to answer that question, Kyle Meyer, who's a postdoctoral researcher in the lab, he set up a pretty epic field experiment where we had 6 blocks, and within each block, we replicated little mini plant neighborhoods. And each of these plant neighborhoods looks like this. It's a ring of plants that we call the neighborhood with one focal plant. And we manipulated who was whom. So in other words, you had a pepper plant in the middle surrounded by bean, or a bean plant surrounded by pepper, or bean surrounded by bean, pepper surrounded by pepper, and so forth. Okay? And we're always focusing on that focal plant in the middle and asking how the microbiome it managed to assemble was affected by its little mini local community. But the other cool thing that we did was we did it in a sort of Dorian Gray style, if you've read that book, where we let the focal plant grow for a month, we then harvested it, and we replaced it with the same species and the same original age of focal plant. So a 1-month-old tomato, pepper, or bean went into the middle, but we allowed the neighborhood to grow up around that focal plant. And we did that 3 times. And another way of thinking about it is the biomass of the neighborhood became higher and higher, but the biomass of the focal plant remained the same. All right. So what did we find? Well, the best way that we ended up thinking about how to visualize the punchline takes a moment to explain. So here are our 3 harvests, so those 3 cohorts, right? This was a low biomass neighborhood, slightly higher biomass neighborhood, much higher biomass neighborhood. And then what is on the y-axis is the R-squared, so how much of the variation in microbiome composition was explained by different factors. So these are, of course, very complex, highly diverse communities. And so we just try to explain how similar or dissimilar 2 communities are and which factors explain that similarity. And so the variables that came out as significant were block, so where spatially in the field you are, that makes sense, neighborhood, and the genotype. Genotype is a way of saying bean, pepper, or tomato. And what we found is that when you look at how much of the variation is explained by each of these variables over time, at first, in that first harvest, when everyone's quite young, the plant genotype, whether you yourself as a focal plant are tomato, pepper, or bean, was by far the most important factor in shaping microbiome composition. So that you could think about it as that plant selection or plant filtering. But through time, the role of the local neighborhood became more and more important until at the end, it actually overtook plant genotype. And you could think about this as mass effects or this sort of push and pull between dispersal and selection. And so we could say that early on, selection was very effective, but eventually selection became ineffective against this constant pool of dispersing organisms from neighbors. All right. And so Kyle followed up on this by taking plants from, those focal plants from the field, taking off all of their microbes using a sonicator and pelleting those microbial communities and spraying them onto plants in the greenhouse. And we just wanted to, in the field, you can't control who's dispersing where, but in the greenhouse, we can do that. So he essentially replicated this experiment, but by manipulating dispersal directly. And then we sampled the plants and asked, how important is your donor microbiome in shaping the microbiome you yourself as a focal plant can assemble? And sure enough, here's just the statistics here. The host species was highly significant. So even if you spray the same microbial community onto pepper and bean, the end result of that community, because of selection, is very different. The previous host species was also very important. So this confirms the idea that the donor plant, where the dispersal pool is coming from, is important in terms of what you can select upon. And then in some cases, this was not overall at all significant, but in the case of bean, we actually even saw like a grand parental effect. So it even mattered who the neighbors of your neighbor were. All right. So for this section, what I want you to take away is that these transmission processes are all important in shaping microbiome composition. And once we've acknowledged that, we can really think about what that means for agriculture, where all of these processes have been disrupted, often purposefully. But once they're there, these microbial communities, that is, of course, not the end of the story. In fact, you could think of it as the beginning of the story, because then through time, through development, these microbiomes are constantly being shaped and reshaped by different factors, including biotic factors and abiotic factors. And so in this middle section of the talk, I'm going to focus on what happens to microbiomes once they're there. And the first story is work led by Rena Debray, who's a graduate student in the lab, PhD student, in collaboration with another professor at UC Berkeley, Tim Bowles, and his graduate student, Yvonne Sokolar. And what Tim and Yvonne had done is set up a huge field trial at UC Davis, where they were manipulating below ground factors and asking how that impacted tomato yield above ground, right? So they manipulated drought, water availability, as well as mycorrhizal associations. So they have a really neat mutant of tomato plants that does not form below ground mycorrhizal associations. And they were interested in how that might interact with drought and or impact yield. But they, of course, were not looking at the microbiome. And so we asked if we could piggyback or be a parasite on their study and sample what was happening above ground. So in other words, asking how these below ground conditions might impact above ground Phyllosphere microbiota. And the short answer is they really do. So you can think of what they've done in the field as stressors. And if you do, then you can think about, this is the wild type plant that has mycorrhizal associations in full water. And this is just overall bacterial diversity. And it's important to say at this moment that for the most part, there are very few general rules across systems in terms of microbiomes, but one of the seemingly general rules is that a diverse microbiome is a good microbiome. There are a few really cool exceptions, but on average, high diversity is considered a positive trait, if you will, of microbiomes, and most dysbiosis or microbiomes associated with disease tend to have lower diversity. And indeed, when we take away water or reduce water, then you see bacterial richness really drop above ground. But similarly, if in full water, if we reduce these below ground mycorrhizal associations, you see a quote dysbiosis or drop in diversity. And when you do the double whammy of the mutant in low water, you see an even further reduction in microbial diversity. But, and this was nice to see that, you know, that the below ground environment does matter in terms of above ground communities. But what really surprised us at the time is that when Rena went in and asked which particular bacterial taxa were being impacted by these factors, you can ask which taxa are enriched in full water, and so therefore depleted in drought, and which ones are actually enriched in drought. And similarly, you can say which taxa were enriched in the wild type tomato and which were enriched in this mutant. And what's really cool is that these are the same ASVs or the same taxa were popping out in both analyses. So in other words, the same bacterial taxa that are enriched in drought tend to be enriched in full water, but when mycorrhizal associations are reduced. And this really surprised us at the time, but what's becoming clear is that mycorrhiza associations below ground have a sort of underappreciated role in maintaining water homeostasis as well. So it's likely that even with full water, if you have a depleted mycorrhizal community below ground, it's essentially as if the plant is growing in drought. And it's pretty cool, I think, that we find that same signature in the microbiome. So the abiotic conditions certainly continually reshape competitive dynamics within the microbiome, as well as interactions between the plant and its microbiome. So certainly if we want to explain the microbiome, we need to take into account the abiotic conditions. Of course, the other part of this, and I've shown you some data about this already, is that the plant itself selects for microbiota, as I showed you in Kyle's neighborhood study. But I'm going to do a little throwback to this paper because I just love it, and I think it's really useful in thinking about evolution or adaptation, which we often don't think about when we're just thinking about these ecological interactions. So Norma Morella, I've already introduced, did this study that's very rich, and I'm just going to show you 2 punchlines now. But she did a study where we took a very diverse tomato microbiome from the field and then experimentally evolved that community on 5 different tomato genotypes in the greenhouse, where every generation we sprayed a plant 6 times, I believe, starting when they were 2 weeks old. And then when they were 8 weeks old, so I think 2 weeks after the final spray, we harvested the plant, collected its microbiome above ground, and then passaged that onto the next generation of that same genotype and did that again. And importantly, these are all independent lines, right? Each of these is an independently evolving line, but it was in an open greenhouse where everything was randomized. And we used this passaging experiment to ask whether microbiomes can adapt to the host plant environment. So as if you're continually applying the same microbial community to tomato, does that tomato-mediated selection change the competitive dynamics and who is there? And the answer is yes. I'm not going to show this data, but the first thing that happened is that through passaging, diversity dropped a lot, as you might expect. But the neat thing is we lost about 90% of our taxa from the rich, original, diverse field community, but it happened in the same way across these independent lines. And the 10 or so, maybe, no, it was more like 25% that was remaining were the dominant microbes on the plant. So we really did select for a subset of the community that grew very well on tomato. And when you look at that in this sort of Bray-Curtis dissimilarity, you can see that these are just 2 axes that essentially mean nothing. It's just a way of parsing apart microbiome diversity. Points that are farther apart means those 2 communities are more dissimilar to one another. But what you can see is that in the first passage here in red, that we started with plants over here, and all I want you to take away from this is that through time, we were moving in a sort of concerted way, all of these independent microbiome communities away from this starting point. And overall, plant genotype mattered, but passaging mattered a lot more. So we really were selecting for this sort of core microbiome of taxa. And then we had to convince ourselves that this was meaningful, that these were somehow well-adapted microbes. And this really presented quite a challenge for us. And this is why I like presenting this study, because if you ever find yourself working on adaptation of complex communities, it's remarkably hard to come up with an adaptation trait. And so this might be useful. So what we ended up doing was what we call a community crashing or a community coalescence experiment, all right? So what Norma did is she combined all of the taxa that we pulled off of plants from that first passage. So you can think of these as sort of acclimated to greenhouse, but not yet selected upon taxa. And we pooled that into one inoculum. We call it the passage 1 inoculum. And then she did the same thing for the final passage. So this is our selected or evolved microbiome. All right. Then we have droplet digital PCR, which allows us to quantify the absolute number of bacterial cells in a sample. And she used not just droplet digital PCR, but live dead droplet digital PCR. So we knew exactly how many live bacteria there were here in this inoculum, how many live bacteria there were here, and she made a 1-to-1 mix of those 2 communities. And then plants were sprayed with either the original microbiome, the evolved or adapted microbiome, and the mix. And the first thing to say is I'm just showing you P1 and P4 here, is that they nicely separated. This was the first time we'd sprayed plants in the same experiment with these 2. So that was a relief that we had indeed really different microbiomes when we started the experiment and when we ended. But now the question is, where does that mixed community fall? And the neutral model, of course, would predict that it should be right in the middle because they got half taxa from this and half taxa from this. But in fact, what we found is that that community crashed, community coalesced inoculum was statistically indistinguishable from the final passage inoculum. And a different way of saying that is that our selected microbiome was now uninvadable by all of those taxa that we had selected out over time. And we thought that was pretty compelling evidence that, that if you give them time, these microbiomes really do adapt to their specific plant hosts. And that being said, I just said specific plant hosts, but we didn't test that here. We only did this on tomato. And we're just wrapping up a large-scale study led by Kyle Meyer, where now we have manipulated the plant species on which they've been evolved. And so look for that in the next couple of months. Sorry, it's a little too early to present it today. I'm going to keep my phone up so I can check time. Yeah, perfect. Okay, so the last little vignette I want to focus on about community composition before we switch to the final part, which is function of the microbiome, was bacteriophages. And this is a slightly older paper now. We have a large new project that's being led by Rena Debray to expand upon this and link it with Norma's previous results. But I'm keeping it in here because often when we think about microbiome composition and what shapes it, bacteriophages are ignored. And I think that's really a mistake, and here's why. So what Norma did in this experiment was the same methods I've been showing you this whole time. We went out to the field, we sampled tomato plants from a natural organic farm environment. But in this case, when we took all the bacteria off the Phyllosphere, we didn't pellet the bacteria as we usually do. Instead, we took the whole leaf wash, so essentially the buffer where we'd shaken off all the bacteria in the Phyllosphere, and we size separated it. So first, we put the leaf wash through a 0.2 micron filter, where we collected all the bacteria and fungi and plant cells, if there were any, on the filter. And we call that our phage depleted community. And actually, just because I don't know everyone on Zoom as well, there might be a broad audience here. Let me catch you up quickly on what I'm even talking about with phage. Sorry, I should have done. These, of course, as you see in the picture over here, are viruses that infect bacteria. And our interest is focused on lytic phages. So these are viruses that infect and kill. They're obligate killers of bacterial cells. And so as they target particular bacterial hosts and kill them, we expect that to change competitive dynamics. Sorry, I should have done that intro first. All right. So in this top community here, we've now washed out any free viruses that were on that leaf when we sampled it. And it's phage depleted, but not absent, because any virus that was actively infecting a bacterial cell would, of course, still remain in that community. Then we took the potential free phage, all of these free viruses here, and we concentrated them in a 100 kilodalton filter. And then we sprayed juvenile plants in the tomato plants in the greenhouse, with either the phage depleted microbiome or by recombining those microbial communities with the phages that we've just separated out from those communities. All right. And the first thing we had to ask is, we had no idea if there were even phages there, right? This is just a fields experiment, not even an experiment, tomato just growing naturally in an agricultural setting. Were there phages there? Did they matter? And here we again use droplet digital PCR to quantify absolute bacterial densities 1 day after we sprayed our plants. So on the left here, you have tomato plants that just received the phage depleted bacteria, and luckily the bacteria grew in 1 day, and they grew to higher densities than when you applied those exact same bacterial communities, but with their phage. So that's saying phages are out there all the time. They're killing bacterial cells all the time. And in this case, during this early community assembly, it's probably a pretty big impact on bacterial density as phages are killing bacterial cells. But does this matter? I mean, surely just because some bacteria are being killed, that niche space is just going to be filled and you'll end up in the same place over time. And here we can think about what evolutionary theory tells us. Yay, theory to the rescue. We have predictions about how phages should be impacting these communities. And the two predictions, the main ones that we're focused on are the sort of, well, what in evolutionary biology is called negative frequency dependent dynamics, and in microbial ecology is called kill the winner, which is the simple idea that through evolutionary time, parasites, bacteriophages, should be evolving to infect common bacterial or host types, right? Simply that's how evolution works. If there's a lot of a particular host around, any parasite or phage that can infect that host will have an evolutionary advantage, be selected for, and as that particular parasite type increases in frequency over time, it kills the winner. It reduces the density of that particular host, opening up niche space for more rare species or genotypes. So all of that is to say, we had a pretty good body of theoretical literature behind us to predict that if this is happening, if there's a parasite there in the community killing a lot of bacteria, it should increase microbial diversity or microbiome diversity. And that's indeed what we found. So if you look here, this is 1 week later. And I should say, after a week, there was no longer a signature on absolute abundance, which kind of makes sense. Everything grew up to carrying capacity, regardless of who was there. But now, where you see the big difference after a week is who won, right? And here you can see that when you recombine these microbiomes with their phage, overall Shannon's diversity, this number of taxa, species richness, was higher in the presence of phage than in the absence. And I still think this is an incredibly neat result and one that should be leveraged in terms of how we move forward and think about microbiome diversity. And we're doing a lot of work on this front, not just in plants, but also in animal models and thinking about how bacteriophages might act as promoters of microbiome diversity. But the other really neat result where we had a little bit less theory to guide us, but makes some sense, is that in the presence of phage, these communities were actually more similar to one another. So let me walk you through what that means. This is the average Bray-Curtis distance or dissimilarity between two plants. So two tomato plants that received exactly the same bacterial inoculum by stochastic chance ended up in different places in terms of their microbiome composition. And that is reduced when phages are there. So you could think about phages as sort of steering the community assembly process so that two plants have more similar microbiomes to one another, which I also think could be really leveraged if we're trying to engineer or gently nudge microbiomes. So I'm going to leave time for questions. Great. So then the final section of the talk is I've convinced you, I hope, that these microbiomes are there. They get there through a variety of mechanisms. They're constantly changing through time, and we're really getting a good feeling for what's shaping their composition, how they're changing. But all of this only matters so much as their function and whether they're actually impactful. So in the last bit here, I'm going to focus on the Phyllosphere as an important factor in shaping plant health and disease. And some of this should look familiar. I've already introduced Shirley and Norma's neat experiment where they took off and sequenced all of the seed microbiota of tomato. But what I didn't tell you at that time is that they then, when they collected these seed epiphytic microbial communities, they did essentially a transplant experiment. So we were following fecal microbiome transplant data with keen interest in humans, the idea that you can take someone's microbiome and give it to someone else through these FMTs. Well, we've been doing the same thing in plants. So what Shirley and Norma did is they took off the epiphytic community of seeds, pelleted that into a microbial inoculum, and then surface sterilized all the seeds. So now we get rid of all of the epiphytic microbes. We grow those seeds in sterile water agar, allow the seeds to germinate. But in some cases, before we did that, we re-inoculated the seeds with the epiphytic microbiome we had just taken off. So now same set of seeds with and without their own seed epiphytic microbiome. And then after germination, once the seedlings were growing healthily, we flooded them with Pseudomonas syringae, the pathogen, and tracked disease progression of bacterial speck. And what we found, these are the 4 different tomato cultivars, if you remember, so biological replicates. And in each of the tomato cultivars, there was an effect of the seed microbiome. And what you're looking at here is through time, how diseased these seedlings became once challenged with the pathogen. And in the tan color, you see the seeds that never had a microbiome, so essentially the sterile-ish seedlings, versus those that had been re-inoculated with their own endogenous microbiome. I'm not going to show this result because it's just too much data, but Shirley did also go in and pick individual Pantoia isolates, and she could show that you don't actually need a very diverse community at all to get these disease protective effects. Really, one taxa of Pantoia was enough to prevent pathogen establishment at this early seedling stage. And the other really surprising result that we found was that, and this is something that we found in numerous studies, all with very different goals, is that more was not necessarily better. So really just throwing these bacteria on there at very high densities did not increase protection, and in some cases actually reduced protection. So there's a lot to learn about how all of this translate. I don't think it's as simple as just soaking all of our agricultural plants in microbiota. That seems to be very counterproductive. There's a really nice level of just enough bacteria to be disease protective, but not too much. Okay, so that original seed microbiome that's vertically transmitted is disease protective. I already introduced Eli's experiment, where he's now expanded that to this synthetic community of adult plants, or at least juvenile plants. Well, we spray them as juvenile plants. Well, in this experiment, unlike Shirley's very early seedling experiment, we actually finished spraying usually around week 6. These are different trials that we ran. We followed plants all the way through to the end of harvest. So we asked how this early microbiome colonization in a greenhouse setting, where we already know they're depauperate of microbiota otherwise, how that impacts plant fitness. All right. So we counted the number of fruits, but we did all sorts of other analyses as well that I won't show you, because what I want to point out here is that across different levels of fertilization, which is what you're seeing in the numbers at the top here, we found that this actually matters, not just to disease protection. There's no disease here, or at least not on purpose. We had one trial that was taken down by powdery mildew, but even in the presence of powdery mildew, what we found is that this early life microbiome colonization leads to higher yield, higher numbers of tomato, and importantly, they're not smaller, which is kind of neat. So there's not we're not exploiting a trade-off here. We seem to be providing more resources. So more fruit of the same size when they get this phyla start. And the other neat thing is that by accident, we fertilized them so much that we really stressed these plants out. All right. And in this really stressful environment where they're overly fertilized and essentially in a toxic environment, then this phyla start microbial community really mattered in terms of tomato yield. And I'm going to stay on that point for a second, because way back many years ago, I had a graduate student, Maureen Berg, who came over from Michael Shapira's lab for a year, and she ran an experiment with a very similar synthetic community to the one I've just presented, but slightly reduced in diversity. And she sprayed adult plants or juvenile plants and then challenged them with Pseudomonas syringae. And indeed, as should come as no surprise by now, when you spray the synthetic community onto plants, either diluted or undiluted, we saw this disease protective effect, less Pseudomonas syringae in the presence of a microbiome. But this result that's bothered me or excited me ever since she published this data is that, long story of how we figured this out, accidents as always, we found that if you fertilize plants, this disease protective effect of the microbiome vanishes. All right. So these are plants that don't receive fertilizer. These are plants that do receive fertilizer. And there, how much pathogen, how much disease was acquired didn't matter based on microbiome status. And this was recently replicated with this exact synthetic community, the Phylostart community by Rena Debray, with an entirely different fertilizer. This is bat guano, not an off-the-shelf fertilizer like we used previously. And we see exactly the same result. That basically when where these plants really rely on their microbiome is when conditions are stressful, when there's low nutrients. And I'm going to stay on that theme for a moment, because we also took the synthetic community and put it onto Arabidopsis accessions that were either diploid or polyploid. And I won't go into some of the punchlines of this study, but I want to say that even though ploidy level didn't impact who colonized the plants, we again found this striking difference in how dependent these plant taxa or accessions were on the microbiome for disease protection. So Arabidopsis is also host to Pseudomonas syringae, so we could follow exactly the same methods I've been describing. And what Eli and Mick Song, his collaborator, found, I didn't put it in here. Well, I'll just tell you then, less data, sorry. What they found is that, actually, you know what? Since I have a moment, I'm gonna find, give me 1 second. That slide must have just ended up at the bottom somewhere. Oh yeah, okay, we'll just jump to there. All right, there it is. All right, here's the data I was just talking about a moment ago to say that we found ploidy level didn't affect microbiome composition overall, so that's fine. But when we challenged the diploids and the polyploids with Pseudomonas syringae, we found that when they get the SYNCOM in red, the diploids had far less disease than the polyploids, than the control diploids. But for the polyploids, they did not rely on the SYNCOM for disease protection. But in this case, Eli and Mick did a step better. They used RNA-seq to look at the transcriptome of the Arabidopsis in the presence and absence of the microbiome after disease challenge. And what they found is that for the diploid, if you look at the genes that are expressed for when they're inoculated with Phylostart versus when they're not, it makes a huge difference. Loads of genes are up and down regulated simply based on the presence of this synthetic community, and that was not true in the polyploids. And to make this even more compelling, if you look in and focus on defense expression genes, those that we know are involved in Pseudomonas syringae infection, what they were able to find is that for diploids, there were these huge changes depending on whether or not the synthetic community was there. And for polyploids, those changes weren't there. And after sort of some twisting in your brain, you can think about how this relates to the fertilizer story and the over-fertilizer story of stress, but also the just general low nutrient environment, which is to say that maybe the polyploids they're able to express these genes with less costs all the time. And so they're essentially in a less stressful environment, but it's a genomic environment than diploids, where these expression trade-offs are probably more pronounced. Maybe, I mean, this is just hypotheses now. But if we think about it that way, then polyploids might not need a microbiome because they can upregulate all these genes all the time, whereas diploids rely more on a microbiome for defense. Okay. So again, to wrap that part up, microbiome-mediated protection is likely to be, well, it is very important, but it's likely to be most important in these more stressful environments. And that's basically soon to be, or already is, all of Earth. So we need our microbes. All right, and so just to wrap up then, I think that, by applying these core, I showed you one example where theory directly translated, but even when it's not so direct, using these core principles and theory that we have in ecology and evolution, we can, as we bring them over to explain microbiomes, we can really better understand microbiome assembly. We can manipulate it. We can begin reshaping or engineering microbiome composition, and in doing so, increasing disease resistance, creating more resilient, drought-tolerant, for example, crops, and overall relying less on fertilizers and pesticides. So I think it's a really fun playground for being an evolutionary biologist with a direct line to impact. Yeah, and with that, I'll take questions. Thank you. Oh, you need a microphone. It's official.
Wow. Thank you for this amazing talk. I like the analogy with population genetics, basically, and community ecology here. And I have one comment and one question. Comment is that when you say that you don't have theory explaining or predicting more similarity between your communities when you have microbiomes, I would say that when you have, when in population genetics, when you have frequency-dependent selection, you also get less variation through geography, for example. And one way of finding a loci on the frequency-dependent selection is to look for loci which have more similarity among populations than average. So I would say somehow it does make sense in this framework.
Yeah, that's great to hear. So this idea that you have lower Bray-Curtis dissimilarity in the presence of phages, that somehow phages are that negative frequency dependent selection is expected to, under population genetic theory, do exactly that. That's great. I would, yeah.
Maybe just a hypothesis.
Yeah, no, I think this makes sense. And then your original comment about how I mean, it is true that these theories were designed at population level with population genetics in mind, negative frequency dependent selection in particular. And I flippantly said that it's the same as kill the winner dynamics in microbial ecology, but of course that is one of the major differences, is the kill the winner theory as originally designed by Thingstadt was focused on explaining species level diversity. But fundamentally, the process is exactly the same. It's parasite adaptation to common host types. And so I do go back and forth between population level and community level ideas very frequently in my own thinking process. And I actually think it's a big challenge for the microbiome field, because bacteria are very strange, right? There's very little sexual recombination, but there's a lot of recombination through, in part, phages and horizontal gene transfer. So whereas in population genetics, individual populations are linked up through genetic exchange, in microbial communities, different species are linked up through genetic exchange, which, which makes things complicated, but also I think gives us an excuse to use population genetic theory.
Okay, so we now have a question online by Martin Guillemet. Martin, you can unmute your microphone now.
Yeah. Can you hear me?
Yes.
Okay. Hi, thank you. So I was wondering, when you talk about the Bray-Curtis distances between the bacterial populations, do you think the difference between the phage-depleted spray and the not phage-depleted spray?
What do I think the difference is?
Could be due to the fact that could it be linked to the fact that when you deplete, when you do the filtration, there's a big bottleneck in the phage populations, which lead to a huge stochasticity and a different differentiation in the phage populations, which in turn leads to the differentiation in the bacteria population?
Okay, interesting. So in other words, I've been thinking about that result where the phage depleted communities are more dissimilar than in the presence of phage. I've been thinking about it as relaxed selection in the absence of phage, but you're arguing that maybe it's actually that there's still very strong phage mediated selection, but just by fewer phage types. Is that fair?
Yeah, I mean, if there are still phages, they're going to grow back. So
yeah. Yeah, that's interesting. And then, but somehow that phage bottleneck would lead to more stochasticity, drive communities further apart. And you're thinking?
Because if there was a bottleneck in the phage population, then maybe phages infecting some bacteria would just go away. And
No, that's an interesting idea. I mean, importantly, the bottleneck would have been the same for all phage depleted communities. So they did receive exactly the same inoculum. So if there was one phage that snuck through, it should be the same phage across. But of course, you're right that if it was at very rare frequencies, it should pop up. No, I'll have to think more about that. That's an interesting idea. And possibly one we could experimentally test quite easily. So in other words, it's not just phage presence absence, but phage diversity that matters here. Yeah.
Okay. Thank you.
Yeah. Thank you. Oh, you have the microphone. Great.
Yeah, I have a number of questions. I'll start with one, and we'll see. I'll ask the rest if there is time. So my first question is on the part where you showed that the microbiome of when you grow plants
Greenhouse?
greenhouse,
Yeah.
the microbiome evolves. And I was wondering what you think is driving this process. And this has to do with the status of the microbiota that get pruned out. So do you think it's the microbiome adapts to the host plant, or do you think it adapts to the greenhouse environment?
Yeah.
And to the ones that get pruned out, you think they just reflect in the wild a sort of sourcing dynamics?
Yeah. Great. So I'm going to answer the questions backwards. So the idea that, of course, through selection, we were indeed pruning out species. I mean, we are doing a very unnatural, sonicating bacteria off plants, pelleting them, freezing in some cases. So we were quite sure that we would be selecting for taxa that also survived our sampling process. So in part, that's why we were so relieved with that community coalescence experiment at the end, where all the things that we had pruned out potentially through experimental design, even when you put them back into the system, they do not outcompete the ones that were selected for. So that was one of the key things about that community coalescence experiment that convinced me anyway that the experimental methods were not the key reason that we found this adaptation. But to your point about how much of the adaptation or selection was from the plant host versus the greenhouse, I think this is a really important question. And we've recently become quite convinced that it's going to have a lot to do with the greenhouse. So without a doubt, these microbes are adapting to a tomato growing in greenhouse conditions. And I think they're at least equally important, those two environments, because now I just said we've finished an experiment where we've done it on different plant species in the greenhouse. And there are certainly taxa that you see concerted change across plant species that suggests some things just grow well in the greenhouse, without a doubt. But nonetheless, we still see these plant genotype effects that I showed you here and plant species level effects that we've seen. So the short answer really is that both. The greenhouse matters a lot as a microbial environment, but the plant genotype or species still matters as a different growth condition. Yeah. Thank you.
Thank you. That was really so much interesting microbiology. I was going back to the question from Zoom, thinking about the same question a little differently, namely the stoichiometry of the phage and the bacteria. It's great that the phage-depleted bacteria grew up so quickly. What would have happened if you just put the not phage-depleted culture right back onto the plants? Instead of separating them and putting them back together, just put the wash pre-filtration back. I would expect you'd also see, at least in day one, the same depression.
Yeah, that's an interesting idea. I mean, the reason we didn't do that is because by filtering, I mean, we have fundamentally changed a lot of things about that bacterial community by filtering. And so we were worried that the leaf wash itself wouldn't have a fair comparator, right? Similar to what we did with the seeds, you have to take the microbiome off first so that everyone gets treated the same, all the seeds get bleached and so forth before you put it back on. If you were to simply compare it to a seed, that hasn't gone through all that process. So I think that was our thinking of using that as a control. But you're right that, maybe that pure leaf wash would have more diversity that we could capture. But then I just don't know what the fair comparator to that would be experimentally.
Yeah. And even just coming at it from what I was thinking about, the stoichiometry, like how many, what's the MOI you recombine as compared to what it was before?
It's a great question. Yeah. And so this is the huge challenge in the field. We do some experiments where we have very specific phages we use. We have genomes of those phages. We can develop primers and probes, and we can quantify their densities. And we've done that a lot. And we've even done that in plants. And we can see how MOI changes through time. And that's a whole nother body of work I didn't present today. But for a whole phage community, unlike bacteria, where you have the 16S marker, as you know, for phages, there's no conserved marker. And I'm just not sure how we would ever be able to get there. We've tried some flow cytometry, where it kind of seems to work a little bit. You can quantify how many virus-like particles are in a sample, but it's
Yeah.
It's certainly imperfect at
Yeah,
best.
The question isn't at all critical. I'm just
No, I think
straight
it's
up curious to know. What the
as am
odds-
I. Yeah, exactly. And especially because we have such, I didn't have time to present them, but we have this recurring weird dose dependence that we keep finding. And I'm wondering, how that plays into the stoichiometry here. It would be incredible to start getting at. But I think all things are pointing to these ecological dynamics or these stoichiometries to be really important, but they're hard to measure, easier to manipulate though. So maybe we start there. Yeah. Thank you.
Hey, Britt.
Yes, hi.
Really nice talk. Everyone seems to be jumping onto your phage parenthesis. This showed that phage is everywhere all the time and it matters. And to what extent do you think would they matter for all the other results that you showed us where phage wasn't there, at least in your results?
Yeah, this is always the challenge when you're doing this field work and then you're trying to bring into the lab and answer more simplistically. You're absolutely right. I will say that the vast majority of our experiments, we do pellet bacteria. And we do that for many different reasons, but mainly because in order to be able to sequence and quantify the bacterial community, you need a bacterial pellet. To be able to get to densities to colonize a plant, you maybe don't need it, but it's helpful. Certainly for storing in the freezer, it's critical. So all of the experiments that we do, for the most part, we deplete phages as part of our processing step, which is really sad. But at least I can say that we accidentally have excluded phages from most of our study. That's not to say they're not important, but we haven't directly looked at them apart from that one study. And then I guess a similar, just to dovetail from that for a second, all microbiome studies, they are all phage studies as well. It's just that it's so often not examined or not observed as part of it. So I think without a doubt, the data are there. And we have actually been working on a big meta-analysis of previous papers from across systems to try to figure out when and how phages are most impactful in microbiomes. So that's the punchline. Even when you're not looking, they're there and they're important. But you can't study everything all the time. So I think just knowing that they're a potential explanatory mechanism in your data set is important, is an okay starting point. Let's put it that way. Yeah.
I think we should keep track of the order to which we passage the microphone, such that we can retrace the route of infection afterwards. I have another non-phage question. You showed us that the physiological state of the plant affects the microbiome. And if I understood correctly, these plants were placed in different physiological conditions from the beginning. Is it known whether a change of the physiological condition during a plant's lifetime is going to affect its microbiome? And perhaps most importantly, at what rate does this change occurs? Is this something very fast or?
Yeah. So the joke in the lab at the moment is if you come to me with a question that starts, does X affect the microbiome? Don't do the study because the answer is yes. Everything affects the microbiome. And that's not, so yes, there are certainly quite a few papers out there from across every system that has looked. The microbiome, it really matters the physiological state of the host, that's for sure. But then your second question of how much does it matter is not particularly well studied. That being said, the best studied would be probably the human gut, of course, where the most work on microbiomes. And there it seems to happen remarkably fast. If you put someone who's a meat eater on a vegetarian diet, maybe someone in the audience knows better than I, but it's like within 24 hours, you see a complete shift of the microbiome. So yes, it happens very fast. We know less, we haven't really done a lot of the temporal series here in leaves, but I would imagine equally. In fact, actually, even more so than these extreme changes, there's beautiful data from across systems of diurnal changes in the microbiome. In vertebrates, because of hormone changes but in plants through day-night cycles, it changes dramatically and in somewhat predictable ways. So yeah, very dynamic systems. And again, interesting, but also really important to think about when you are designing your experiment, when to sample really matters and how you're comparing across your individuals. These are big decisions.
One last small question.
I will try to make it small. Thank you very much for the presentation. So I begin by the final question, and then I explain how I came to this question. So the final question is, what is the effect of the bacterium on the plant in terms of competition between bacteria, right?
Okay.
What is the direct effect of the bacteria competition? Versus the state of the plant. And the question comes from the fact, from your experiment, when you did look at the plant capacity to resist to Pseudomonas when it was with the seeds, were with the microbiome or without.
Okay.
Because I presume the inoculation of the seed did influence the total microbiome
yes.
of the plant, and not only the rhizosphere, I presume, because Pseudomonas, you did look at the Pseudomonas is on the Phyllosphere, right?
Yeah.
So then, what is the impact of the microbiome on the health of the plant? Because maybe what you see is only the health of the plant and not a direct competition.
Okay, so that's a good question, and I'll answer it in two ways. One, very specifically in the seed project, Shirley was the most meticulous undergraduate. She really probed deep. And one of the things that she did was included a heat-killed inoculum control. So we inoculated seeds with Pantoia, when she went down to these single species levels, with Pantoia at different doses, and then with the heat-killed Pantoia culture. And could show that heat-killed Pantoia also was changing the plant defense to Pseudomonas syringae, but not as much. So another way of saying that is that the disease protective result is certainly a combination of both changes to the plant immunity as a result of the bacterial signaling and direct competition. And to build on that, with the synthetic community, we're able to do, really probe deep on who's competing with whom. And we can show that through a new set of models we've been working on with Marline Bruning, that when we use these metabolic models based on biolog profiling for each of the individual bacteria, that it really matters. Niche overlap is quite important. So some things that you predict to be very high competitors of Pseudomonas syringae are indeed the most competitive, but not always, which is equally interesting. So I think the somewhat boring answer is it all matters. The direct competition matters, but there's also an interplay with the hosts through signaling and changes in defenses. Yeah, thank you.
Well, thank you to everyone who asked questions in the room and on Zoom. And thank you very much, Britt, for this great talk.
Yeah, thanks for having me. Nice to see you all.